इम्यूनोलॉजी का एक छोटा टॉपिक के बारे में, जो कि हाउ इंटरफेरॉन सेव्स आवर बॉडी फ्रॉम वायरसेस। ओके, और अभी इस कोरोना वायरस का सिचुएशन में ये सबसे इम्पोर्टेंट है। तो आपका क्या हाल है लाइक इंटरफेरॉन को लेके, और इंटरफेरॉन को लेके आपका क्या कॉन्सेप्ट है? आईआईटी जैम हो या फिर मानो कि आईआईटी जैम हो, जेएनयू हो, कोई भी एग्जाम हो जाए, interferon is a very important concept and how it is giving protection against viruses. So definitely you can see a lot of viruses and bacteria, they kind of hit our body all the time, right? And in that situation our body fights back and our body's immune system fight back. But for bacteria it's pretty much well known what happens, but for viruses it's less understood how our body can fight back against viruses. तो हम लोग वही देखते हैं, ठीक है। तो पहले हमारा इम्यून सिस्टम को तो हम लोग एक डिफेंस सिस्टम ऑफ आवर बॉडी से कंपेयर कर सकते हैं, है ना। इम्यून सिस्टम को डिफेंस सिस्टम ऑफ अ कंट्री से कंपेयर कर लेंगे हम लोग। तो देखो, इम्यून सिस्टम में भी तरह तरह का डिवीजन है। जैसे एक आर्मी में डिवीजन होता है, जैसे लैंड ट्रूपर्स, एयरफोर्स, नेवी एंड ऑल ऑफ दीज थिंग्स। एंड द वे these navy, air force interact with each other is via walkie talkie, but the cells interact with each other in the immune system via cytokines. And one type of cytokine is actually what? Actually, one type of cytokine is interferon, right? Now, interferon is one of the family of 6 major cytokines. ठीक है। Now, interferon में भी 3 different subfamilies हैं। interferon class 1, class 2, class 3। interferon class 1 जो family है ना, ये सबसे ज़्यादा important है इन्फ्लामेशन, एंटीवायरल रिस्पॉन्स इसके लिए, ठीक है। इंटरफेरॉन क्लास 2 जो है, इंटरफेरॉन गामा, IL-10, ये सारे टी सेल का रिस्पॉन्स को मॉड्यूलेट करता है। और इंटरफेरॉन क्लास 3 रिसेंटली डिस्कवर हुआ है, वो है वेट सॉरी, so somebody joined, so hi Dev Krishna, hi, and also good evening. So guys, this is the continuous playlist of virology where we are talking about several aspects of virus and everything. So definitely it would cover the whole syllabus of IIT-JAM or JNU or any MSc level entrance, even till CSIR net it would cover, okay? And not only that, it would give you update about this coronavirus and all of these things. So this, we would put all of these knowledge from that context, okay? Okay guys, so we're going to talk about interferons today, how interferons are protecting our body. So let's say this cell in the left hand side is getting infected by the virus, but the cell nearby who is there, who is still not infected. So what happens? The cell which is infected, that cell sends the alarm signal, and this alarm signal is nothing but interferons. So interferons are the alarm signals, okay? So let us see, as the name suggests, interferons. So interferons means as if interfere with something, right? Okay, so now what happens? These interferons binds to specific interferon receptors. On what? Interferons bind to specific interferon receptors on the nearby cell. Now the nearby cell get the protection by interferon mediated signaling. तो वो थोड़ा डिटेल में देखते हैं कि इंटरफेरॉन मेडिएटेड सिग्नलिंग कैसे काम करता है। तो पहले अच्छे से देखते हैं। मानो कि हमारा जो बैरियर है, जो स्किन हमारा बैरियर है, राइट, विच इज वेरी कंपेरेबल टू अ बैरियर ऑफ अ स्टेट। नाउ कोई टेररिस्ट आ गया बैरियर तोड़ के, then our patrolling police officers would definitely catch hold of them. So our dendritic cells, macrophages, are all those patrolling police officers. Now macrophages, dendritic cell, all of them have dedicated toll-like receptors which can detect viral dsRNA or single-stranded RNA, viral DNA, etc. So viral DNA can be detected by these police officers. I mean, these patrolling police officers, which we are calling as antigen presenting cells, the macrophages and the dendritic cells. Okay. Now, what happens is like these toll-like receptors, especially the intracellular toll-like receptors, such as TLR3, which would detect the presence of viral dsRNA, TLR7, which would detect viral single-stranded RNA, or TLR9, which would detect viral DNA signatures. So these things detect all the viral or presence of a virus inside a body or invasion of a virus inside the body, and then it takes all the necessary measure to alarm the body how to fight back. Okay, so toll-like receptor, which is a part of the innate immune system, can really fight back. Okay, so then what happens, these dendritic cells go and activate other cell types such as T helper cells. In this case, this T helper cell is a cytotoxic T cell. Now, other cases, the T helper cells, I mean, the cytotoxic T cell can be activated, the T helper cells can be activated, both the kind of situation can happen. Okay. And if it's a T helper cell, it would also activate a B cell to become a plasma cell. And it can secrete antibodies against that particular virus, opsonizing that virus or making that virus weak. Right? So if the virus is totally coated by the antibodies, then what would happen? That the virus cannot interact with all the receptors present on the cell membrane. As a result, it cannot sort of infect that particular cell, and also the macrophages, the dendritic cell all have the FC region receptor. The FC receptors would now engulf that virus, and after engulfing, what would happen? They would take them inside. Okay. Now, defensive role also is played by CD8 positive T cell, especially against viruses. How? Let's say this particular cell is infected. Okay, this particular yellow cell is infected by virus. Okay, now what would happen? This virus would try to, if it's a RNA virus or a positive sense RNA virus, so do let me know if you know positive sense or negative sense RNA viruses or not, or otherwise check the introductory course in the same page, in same Unacademy page. In the YouTube page, we have an introductory virology series where I talked about what are positive sense RNA, what are positive sense RNA, what are negative sense RNA, and all of these things. Anyway, the virus can create proteins, and some of this viral protein by mistake would be degraded as well, and then these proteins would be displayed on the top of the cell by class 1 MHC molecule. Okay, so these proteins would be loaded onto the class 1 MHC molecule. Any cell generally displays its self antigen on the surface. Okay, but in this case, since the virus is present, sometime by chance viral protein can also be depicted on the top of the on this cell membrane, infected cell membrane. That is an alarming signal for the T cytotoxic cell. Now the T cytotoxic cell would understand that okay, something is going wrong because this particular cell should not be displaying a viral antigen, right, or viral protein. So something might be wrong. In this situation, what would happen is the CD8 positive T cell would secrete granzymes and perforins. Now these granzymes and perforins would evoke apoptotic pathway, right? So let's see this process in bit more details. So granzymes are inside and perforins create pore in the membrane such that the granzymes get in. Once the granzyme get in the vesicle, they come out in the cytoplasm. Okay. Now granzymes create leak on the membrane of the mitochondria. Now several pro-apoptotic protein like bead, BAX, etc. are already there. So they are actually augmented by these granzymes. Now cytochrome C gets leaked out, which is supposed to be present as a component of the electron transport chain. It gets leaked out in the cytosol. Now several proteins such as APAF1 detect that and ultimately help to cleave inactive version of the caspase to an active version of the caspase. Okay. So whole point is like granzyme and perforin activate caspases, and caspases are the executor for death pathways, right? Similarly, these cells also express Fas ligand and Fas receptors. So these also can activate other caspases, which ultimately boil down to caspase 3 activation, and thereby what happens is apoptotic pathway gets evoked. Now this particular cell which was affected by the virus is sort of dead, but the virus already has replicated in that time, and virus can infect other cells. But how does body ensures that that kind of mechanism does not happen? That is kind of ensured by interferons. The cell which is dead, it cannot save itself, but it can secrete now interferon. So it would secrete interferon to all of these nearby cells, and the nearby cells, after getting interferon from the virus affected cell, they start producing antiviral mechanisms. Okay, so after producing antiviral mechanism, what happens? That interferon try to blocks the viral assembly process or the viral replication process. So there could be different signal which is orchestrated mainly by JAK-STAT pathway. Several stat can be used for the heterogeneity of the response. In this case, a different interferon when binds, a different stat, a different combination of stat is expressed, and that gives rise to, and that can bind to a different region in the gene and gives rise to different proteins or different antiviral molecules. So let's look at the antiviral molecules. First type of antiviral molecules are protein kinase R, which actually sequesters viral dsRNAs and inhibits its translation. So it interferes with the translation process. If virus cannot translate inside the host cell, then what would happen? It cannot create its matrix protein or capsid protein, so it cannot assemble in futures. That's a good part. Then other cases, what happens is like elongation factor 2A also inhibits. Elongation factor 2A is important for translation process. Now in this situation, if protein kinase R phosphorylate elongation factor 2A, then the translation process would be inhibited because elongation factor 2A under phosphorylation condition is getting inhibited. As a second mechanism, other enzymes such as 2,5 oligoase synthase can also block these viral dsRNA, or it can sequester such that it would be cleaved by several RNase molecules such as RNase L. There are other proteins such as MX proteins which sort of inhibit viral capsid assembly and thereby protects the body from virus infection. So pretty much we understand that how our body got saved from this virus infection. So now let's try to look at what happens in case of coronavirus. So in the coronavirus, we see death rate, we see infection rate, but also we are seeing some amount of recovery rate. Quite a lot of people are recovered. So we try to understand that how our body is fighting back against coronavirus and how our body can get recovered. So coronavirus would get inside the lungs, and especially it would affect your alveoli, and you know that alveoli are the air filled sacs where gaseous exchange of air takes place, right? So in case of coronavirus infection, it creates a huge inflammation in the alveolar cells, and as a result what happens, this inflammation results in fluid accumulation and thick mucus accumulation in the bronchi. Now this fluid accumulation or accumulation of pus happens that sort of interferes with the gaseous exchange. So there is always a shortness of breath and breathing problems that is happening inside your body when you're affected with the coronavirus. Now you understand why there is a problem with the breathing, right? But anyway, the cells inside the lungs are also near vicinity with the blood vessels. So T cytotoxic cells would immediately try to hit them, and they would try to kill them, right? Now the cells would be killed by the T cytotoxic cells, but the nearby cells would be safe from infection by the help of interferon signaling. So the dying lung cell would secrete interferon, and now the interferon would create antiviral defense response such that your body is now protected against this particular virus. Now that is why the people who were affected and now recovered, their plasma is valuable. You can isolate